Plants have fascinating ways of creating new individuals, ensuring their survival and propagation. In this video, we'll explore the various methods by which plants reproduce, from asexual to sexual reproduction, and how environmental factors and human influence play a role in this essential aspect of plant biology. Many plants reproduce asexually, in addition to sexual reproduction. In asexual reproduction, new plants are generated from a single parent plant without the involvement of gametes or fertilization. This method allows for the production of genetically identical offspring to the parent plant. Asexual reproduction is advantageous in stable environments, as it does not require the resources needed for flower production, pollination, or seed dispersal. Various types of plant parts are involved in asexual reproduction. Some plants produce new individuals from specialized plant parts like stems, roots, or leaves. For example, strawberries produce runners or stolons which develop into new plants upon contact with soil. Others form specialized structures like bulbs, corms, or tubers containing nutrients and genetic material to support the growth of new plants. Plants can also reproduce asexually through apomixis, where seeds are produced without fertilization. This process involves the ovule, or part of the ovary, giving rise to a new seed, resulting in genetically identical offspring. Advantages of asexual reproduction include faster maturation of offspring and increased sturdiness compared to seedlings. Asexual reproduction can occur naturally or through artificial means, such as grafting, cutting, layering, and micropropagation. In natural asexual reproduction, plants utilize strategies like budding, adventitious roots, or runners to self-propagate. Some plants, like the sweet potato, produce runners that give rise to new plants. Others, like Bryophyllum and Kalanchoe, have leaves with small buds that grow into independent plants when detached. Artificial methods of asexual reproduction are often used to create new plant varieties. Grafting involves attaching a desirable plant stem, scion, onto a rooted plant, stalk, to combine favorable characteristics. Cutting involves planting a portion of the stem in soil to develop roots and form a new plant. Layering involves burying a part of the stem to encourage root formation and subsequent growth. Micropropagation, or plant tissue culture, allows for the rapid propagation of plants from a single specimen in a controlled laboratory environment. The lifespan of plants varies, with some living for only a few weeks, annuals, to others living for thousands of years, perennials. Factors like genetics, environmental conditions, and reproductive strategies influence the longevity and reproductive success of plants. Sexual reproduction in plants involves the fusion of gametes from two parent plants to produce offspring. Unlike asexual reproduction, which produces genetically identical offspring, sexual reproduction introduces variation, which is crucial for species adaptation to changing environments. The process of sexual reproduction begins with DNA replication, ensuring that genetic information is passed on to the next generation. While asexual reproduction results in the formation of identical clones, sexual reproduction facilitates the creation of variants, which is essential for adaptation. In sexual reproduction, two different gametes, each containing half the chromosome number of the parent organism, fuse to form a zygote. Gametes are produced in specialized reproductive structures, such as flowers in plants. These structures typically consist of male and female reproductive parts, the stamen and pistil. The stamen, the male reproductive part, comprises the anther and filament. The anther produces and stores pollen, while the filament supports the anther. On the other hand, the pistil, the female reproductive part, consists of the stigma, style, and ovary. The stigma receives pollen, the style connects the stigma to the ovary, and the ovary contains ovules where seed formation occurs. Flowers can be unisexual or bisexual, depending on whether they contain only male or female reproductive parts or both. Pollination, the transfer of pollen grains from the anther to the stigma, is necessary for fertilization. Pollination can occur through self-pollination, where pollen is transferred within the same flower, or cross-pollination, where pollen is transferred between flowers of the same or different plants. Pollination is often facilitated by pollinators such as insects, birds, wind, or water. Once pollen grains reach the stigma, they release male gametes, which fuse with the egg and the ovule to form a zygote. This process, known as fertilization, leads to the development of an embryo, which eventually matures into a seed. The ovary develops into a fruit, enclosing the seeds. Overall, sexual reproduction in plants involves a complex series of events, from the production of gametes to pollination and fertilization, ultimately resulting in the formation of seeds and fruits, which ensure the continuation of plant species. Next, we'll discuss the alternation of generations life cycle in plants. Alternation of generations means that plants alternate between two different life stages, or generations, in their life cycle: a haploid stage called gametophyte and a diploid stage called sporophyte. The terms haploid and diploid refer to the number of chromosomes contained in the cells. There are two main phases, the gametophyte phase and the sporophyte phase. The gametophyte phase is haploid, meaning each cell contains only one set of chromosomes, N. Gametophytes produce gametes, sex cells, through mitosis. These gametes include sperm cells and egg cells. In the life cycle of plants with alternation of generations, the gametophyte is the dominant phase in bryophytes, mosses, liverworts, and hornworts. The sporophyte phase is diploid, meaning each cell contains two sets of chromosomes, 2N, one from each parent. Sporophytes produce spores through meiosis. These spores are reproductive cells that give rise to gametophytes. In the life cycle of plants with alternation of generations, the sporophyte is the dominant phase in vascular plants, ferns, gymnosperms, and angiosperms. Mosses, ferns, and angiosperms are examples of plants that exhibit alternation of generations. Mosses exhibit a prominent gametophyte phase, where the leafy green structure commonly seen represents the gametophyte generation. The sporophyte phase of mosses is much smaller and dependent on the gametophyte for nutrition. Ferns have a dominant sporophyte phase, with large, leafy fronds representing the sporophyte generation. The gametophyte phase of ferns is typically small and inconspicuous, often living underground or on the undersides of leaves. Angiosperms, the most diverse group of plants, exhibit alternation of generations with a dominant sporophyte phase. The gametophyte phase of angiosperms is highly reduced and exists within the reproductive structures of the flower, such as the pollen grains and ovules. Environmental factors play crucial roles in influencing plant reproduction. These factors include temperature, light availability, moisture levels, soil nutrients, and the presence of pollinators. Each of these factors can impact different stages of the reproductive process in plants. Temperature affects the timing of flowering, pollen development, and seed germination in plants. Extreme temperatures, either too hot or too cold, can disrupt flower formation, pollen viability, and fertilization. Light is essential for initiating flowering in many plant species, as it serves as a signal for the transition from vegetative to reproductive growth. Photoperiod, or the duration of light and darkness in a day, can influence the timing of flowering in plants, particularly in those with photoperiodic responses. Adequate moisture levels are necessary for pollen tube growth, fertilization, and seed development. Drought conditions can hinder reproductive success by reducing pollen viability, inhibiting pollen tube elongation, and causing premature flower abortion. Plants have evolved various mechanisms to adapt their reproductive strategies to changing environmental conditions, ensuring reproductive success even under adverse circumstances. These adaptations include adjusting flowering time in response to environmental cues such as temperature and photoperiod. Short day plants, long day plants, and day neutral plants exhibit different flowering responses based on the duration of light exposure. Some plants rely on wind pollination, while others depend on animal pollinators such as insects, birds, or mammals. Plants may produce attractive flowers, nectar, or fragrance to attract pollinators, enhancing the chances of successful pollination. Plants employ various methods to disperse their seeds over long distances, including wind dispersal, water dispersal, and animal dispersal, for example, through ingestion and transportation by animals. Seed dormancy and germination timing may be influenced by environmental cues such as temperature, moisture, and light availability. Overall, plants exhibit remarkable plasticity in their reproductive strategies, allowing them to thrive in diverse environmental conditions and ensure the continuation of their species. Human activities have significant impacts on plant reproduction, influencing both natural ecosystems and agricultural practices. Cultivation practices, such as land clearing, irrigation, and fertilization, can alter natural habitats and disrupt the reproductive cycles of native plant species. Hybridization, the deliberate crossbreeding of different plant varieties or species, is commonly practiced in agriculture to develop new cultivars with desirable traits such as disease resistance, increased yield, or improved flavor. Genetic modification techniques, including genetic engineering, allow scientists to introduce specific genes into plants to confer desired traits, such as herbicide tolerance or insect resistance. While human intervention in plant reproduction has led to significant advancements in agriculture and food production, it also raises ethical considerations and implications. Concerns about the environmental impact of genetically modified organisms, GMOs, include potential harm to non-target organisms, unintended gene flow to wild populations, and the development of herbicide-resistant weeds. Additionally, there are ethical concerns regarding ownership and control of genetic resources, as well as issues related to biodiversity conservation and the protection of traditional farming practices. As technology continues to advance, it is essential to carefully consider the ethical implications of human intervention in plant reproduction, and to prioritize sustainable and environmentally responsible approaches to agricultural innovation. Collaboration between scientists, policymakers, and stakeholders is crucial to ensure that plant breeding practices uphold ethical standards, promote biodiversity conservation, and contribute to global food security in a responsible manner. Understanding the mechanisms of plant reproduction is essential for both scientific knowledge and practical applications in agriculture and conservation. From the intricate processes of pollination and fertilization to the complex life cycles of plants, the study of plant reproduction offers insights into the diversity and resilience of plant species. Throughout history, humans have played a significant role in shaping plant reproduction through cultivation, hybridization, and genetic modification. While these interventions have led to remarkable advancements in crop breeding and food production, they also raise important ethical considerations regarding environmental sustainability, biodiversity conservation, and equitable access to genetic resources. Moving forward, it is imperative to balance the benefits of human intervention in plant reproduction with ethical considerations and environmental stewardship. By promoting sustainable agricultural practices, conserving genetic diversity, and prioritizing the protection of natural habitats, we can ensure the continued resilience and adaptability of plant populations in the face of environmental challenges. Ultimately, the future of plant reproduction lies in our collective commitment to responsible stewardship of our planet's botanical resources. Through collaboration, innovation, and a deep respect for the natural world, we can work towards a future where plants thrive, ecosystems flourish, and humanity coexists harmoniously with the tapestry of plant life on Earth.